Hi everyone, I will discuss about the process of mitosis and meiosis. The process of mitosis and meiosis. Mitosis. Mitosis is a process of nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. During cell division, mitosis refers especially to separations of the duplicated genetic material carried in the nucleus. Mitosis is conventionally divided into 5 stages, known as prophase, prometaphase, metaphase, anaphase, and telophase. While mitosis is taking place, there is no cell growth and all of the cellular energy is focused on cell division. During prophase, the replicated pairs of chromosomes condense and compact themselves. The pairs of chromosomes that have been replicated are called sister chromatids, and they remain joined at a central point called the centromere. A large structure called the mitotic spindle also forms from long proteins called microtubules on each side or pole of the cell. During prometaphase, the nuclear envelope that encloses the nucleus breaks down, and the nucleus is no longer separated from the cytoplasm. Protein formations called kinetochores formed around the centromere. The mitotic spindle extends from the poles and attaches to the kinetochores. During metaphase, the microtubules pull the sister chromatids back and forth until they aligned in the plane, called the equatorial plane along the center of the cell. During anaphase, the sister chromatids are separated simultaneously at their centromeres. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. Anaphase ensures that each daughter cell receives an identical set of chromosomes. Finally, during telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. The chromosomes begin to uncoil, which make them diffuse and less compact. Along with telophase, the cell undergoes a separate process called cytokinesis that divides the cytoplasm of the parental cell into daughter cells. Meiosis. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half and produces four gamete cells. This process is required to produce egg and sperm cells for sexual reproduction. During reproduction, when the sperm and egg unite to form a single cell, the number of chromosomes is restored in the offspring. Meiosis begins with the parent cell that is diploid, meaning it has two copies of each chromosome. The parent cell undergoes one round of DNA replication followed by two separate cycles of nuclear division. The process results in four daughter cells that are haploid, which means they contain half the number of chromosomes of the diploid parent cell. Meiosis has both similarities to the differences from mitosis, which is a cell division process in which a parent cell produces two identical daughter cells. Meiosis begins following one round of DNA replication and cells in the male or female sex organ. The process is split into meiosis and a meiosis II, and both meiotic division have multiple phases. Meiosis I is a type of cell division unique to germ cell, while meiosis II is similar to mitosis. Meiosis I, the first meiotic division, begins with prophase I. During prophase I, the complex of DNA and protein known as chromatin condenses to form chromosomes. The first of replicated chromosomes are known as sister chromatids, and they remain joined at a central point called the centromere. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side or pole of the cell. Between prophase I and metaphase I, the first of homologous chromosomes form tetrads. Within the tetrad, any pair of chromatid arms can overlap and fuse in a process called crossing over or recombination. Recombination is a process that breaks, recombines, and rejoins sections of DNA to produce new combinations of genes. And metaphase I, the homologous pairs of chromosomes align on either side of the equatorial plate. Then, in anaphase I, the spindle fibers contract and pull the homologous pairs, each with two chromatids, away from each other and toward each pole of the cell. During telophase I, the chromosomes are enclosed in nuclei. The cell now undergoes a process called cytokinesis that divides the cytoplasm of the original cell into two daughter cells. Each daughter cell is haploid and has only one set of chromosomes, or half the total number of chromosomes of the original cell. Meiosis II is a mitotic division of each of the haploid cells produced in meiosis I. During prophase II, the chromosomes condense and the new set of spindle fibers forms. The chromosomes begin moving toward the equator of the cell. During metaphase II, the centromeres of the third chromatic align along the equatorial plate in both cells. Then, in anaphase II, the chromosomes separate at the centromeres. The spindle fibers pull the separated chromosomes toward each pole of the cell. Finally, during telophase II, the chromosomes are enclosed in nuclear membranes. Cytokinesis follows, dividing the cytoplasm of the two cells. At the conclusion of meiosis, there are four haploid daughter cells that go on to develop into either sperm or egg cells. Thank you so much, and thank you for watching and listening.